so each of the courses that does research in MCDB is represented here. There are 5 courses, and each of those courses is represented by 1 to 4 students who are going to describe their work. We're going to start with phage genomics, and we're going to have a talk called Identification and Characterization of Novel Mycobacteriophage in Contribution to the World of Microbiology, presented by Xander Graham, Kyla Hinton, and Gabriella Knobs.
Good evening, everybody. My name is Xander Graham, and along with Kyla Hinton and Gabrielle Knopf, we will be presenting the Phage 1161 lab and the research of that. So for this lab, what we looked into was the processes involved in identifying novel bacteriophage. And we're going to get into more detail on that soon. But first, we're going to go over what bacteriophage are and our specific research so you understand how our lab fits into the bigger picture. Alright, so here is an electron microscope image of 2 different bacteriophages located here and here. These bacteriophages were isolated in our lab by Kelsey Jones and Dakota Kisling. So what a bacteriophage is it's a phage or virus capable of infecting bacteria. The prefix micro simply means that it infects mycobacteria, which is just a specific genus of bacteria. So this is an artist rendering of bacteriophage. The important components to take note of are the capsid head, which holds the genetic information of the phage, either DNA or RNA. Also the tail fibers, which recognize specific bacteria and then bind to them. And then finally the base plate, which once the tail fibers have bound to a bacteria, they create a pathway for the genetic material to travel through the phage and into the host bacterium. So once the DNA of a phage has entered a bacteria, which is shown here, phage and bacteria, it can undergo 2 separate life cycles. The first one is the lytic life cycle, which is more common. So once the phage DNA enters the host bacteria, enzymes prevent the bacterium from replicating its DNA and from transcribing its proteins, and instead force it to create phage DNA and phage protein. The bacteria is then forced to assemble the phage inside of itself, and after a certain number have been made, they lyse the cell, which kills it and releases them into the environment to infect more bacteria. The lysogenic life cycle has 2 separate pathways. The first one is the lytic cycle, which I just went over and is shown here. But they are unique in that they have another pathway they can go under. Once the DNA has been inserted inside the bacterium, they can actually attach their DNA to the bacteria's DNA. This creates a prophage, so now the bacteria chromosome has phage DNA in it. When this bacteria replicates, it will produce identical copies of its DNA, which now have phage viral DNA in it. So any daughter cell will also have a prophage in it. Once a specific environmental stressor occurs, such as a change in temperature or the presence of a protein, the prophage inside the cell will switch from its dormant lysogenic state and instead go straight into the lytic state. So after time, it will just decide to flip over and eventually produce more phage and lyse the cell. During our research, we used Microbacterium smegmatis as a host for our phage. This was because it's non-pathogenic, so it poses no harm to us, but it's closely related to several types of bacteria that are extremely pathogenic, so it serves as a great, safe model organism. Some of the more pathogenic ones you've probably heard of are M. tuberculosis, which causes tuberculosis, M. avium, which is of particular concern to people with compromised immune systems, M. ulcerans, which causes ulcerative lesions, and finally M. leprae, which causes leprosy. And I know what you're all thinking, that is not a picture of a bacteria, that is a picture of a really cute armadillo. And you'd be right, but it turns out armadillos are really susceptible to leprosy, so just keep that in mind the next time you go out and you hug your local wild armadillo. And then with that, I'll turn it over to Kyla.
Hi guys, my name is Kyla, and I'll be going over the methodology methodology and research section, or results section of our presentation. So at the beginning of this semester, everybody in this lab was tasked to go out and collect a soil sample from which they were hoping to isolate phage from. If they were, they would be able to create a plaque assay plate that would look like this. A plaque this is an example of a plaque. So a plaque is a usually circular clearing in the bacterial lawn that shows that phage have infected the bacteria and lysed the cell. In this picture, I wanted to show the difference between lytic and temperate phage by plaque examination. So at this stage in our research, we are able to determine if our phage is lytic or temperate based on the plaque picture. So on the plaque picture to your left is an example of a lytic phage. So here we can see that each of the plaques are clear and they're small, which shows that the phage are able to infect the bacteria, recreate phage and then lyse the bacteria. And in the plaque picture on the right, we're able to see examples of temperate phage. You can see here because the plaques are very turbid, you can tell that they're temperate because the plaques are cloudy. And there are two different ways that you can see that the plaques are temperate, one of which they'll either have a halo, as you can kind of see in some of the plaques here, or they will be cloudy, as you can also see in some of the plaques here. Continuing on, the second half of our lab was to characterize and identify our phage through DNA identification and morphological analysis. So the first picture we have here is an electron microscopy, which Xander already showed an example of. This is a magnification of the phage itself. As you can see here, that's the head or the capsid that Xander was talking about, and that's the tail as well that Xander was also talking about. From there, we were able to isolate the DNA from our phage and perform a restriction digest on it. What happened here was we took enzymes and we digested the DNA with that, and those enzymes created cuts in the DNA based on a certain amount of base pairs, which allowed us to run a gel and create bands like this. And from the bands, we were able to create hypotheses about which cluster or further identification of our particular phage. So almost everybody in our lab was able to get to this step, and all of these images are pictures from, are images from people in our lab, so this is our research. Okay. Well, I guess it's, oh great, it's back, perfect. So, continuing on from that, once you're able to hypothesize a cluster or subcluster that our phage belong to, we ran a PCR gel, and from that we were able to either disprove or prove our hypothesis. So as we can see from this PCR gel, there is a positive control, a negative control, and an F1 primer. The positive control and the F1 primer together confirm that the people who ran this gel, Sean Butler and Darren Swift, good job guys, they correctly hypothesized which cluster their phage belonged to. So why does this matter? Over the course of this semester, all of us have been gathering this information in hopes of adding it to a phage database like this shown above. And I want to kind of go over our statistics just to make this process more real to all of you guys who did not participate in this with us. So as we can see from the titer, which the titer is showing the concentration of phage on our high titer plate. And the titers for our semester lab range from 1.18 times 10 to the 4th plaque forming units per milliliter and 1.8 times 10 to the 13 plaque forming units per milliliter, which is a good range. And after that, everyone in the lab, or almost everyone in the lab, was able to classify the morphology of their phage. And phage come in 3 flavors, either myroviridae, podobiridae, or symphoviridea. And symphoviridea was the only morphological characteristic that we were able to assign. This is not uncommon because symphoviridea is the most common type of phage. We can also see the percentage of lytic and temperate phages, and that's shown here. And it's surprising because actually the majority of the lab were able to isolate lysogenic phage or temperate phage instead of lytic phage. In this last graph, we're able to see the clusters and subclusters that we assigned during this lab. And as you can see, F1 and A1 were the most dominant. And now I turn the time over to Gabby.
Hi guys, I'm Gabby, and now that we know a little bit about bacteriophages and why they're important and what we did with them in our lab, I'm going to go over a little bit of how we can apply our research to the larger scheme. So I looked into 2 areas of research. We can apply phage research to the food safety and sanitation area with produce,
and we can apply it to the medical world. So first off, we need to have a reason of why we do phage research. There's a big problem in our medical world right now in our modern age that is called antibiotic resistance in bacteria. This can also be called multi-drug resistant bacteria, and this is due to our overuse of antibiotics. We are using the same types of antibiotics over and over and over, and the bacteria that have mutations that allow them to be resistant to our bacteria causes those bacteria to survive, and therefore they're more prevalent in our bacterial population. So the ones that are dangerous and can cause infection in humans, those are the ones that are surviving and being more present in our world. So on this graph, the blue line, these 3 lines are examples of infectious and dangerous pathogens to humans that have risen in their resistance to our current antibiotics, which is a huge problem. So first off, we can apply phage therapy to the food industry. We have a current issue called, in E. coli, of the E. coli bacteria in romaine lettuce. And we've had a lot of recalls on this produce, and E. coli is a super infectious bacteria that's dangerous to humans. So phytopathogens is actually the name that we call bacteria that infect plants. And there's actually a phage cocktail or phage therapy called agrophage that we can apply to crops to treat these agricultural crops and kind of eliminate these issues of having to recall all this produce. The second application is phage therapy in medicine. We can actually use our antibiotics currently and some new approaches of phage therapy to combat these antibiotic-resistant bacteria. In the graph here, you can see a specific type of phage was used to treat a non-antibiotic resistant bacteria. And we can see here, the blue line is just the bacteria by itself in cell density, so it grows just on its own. And then the 3 other lines, the green, red, and yellow, those are instances where we apply antibiotics or phage therapy. So this was very successful, and we can see here that we can actually use phage therapy in a substitution of antibiotics, and it's just as successful. So this eliminates, we can actually use phage therapy and antibiotics together to eliminate the possibility of temperate phages, like Xander and Kyla talked about before, which eliminates the possibility of bacteria sticking around that still have phage therapy or phage DNA inside of them. Okay, thank you so much for listening.
Antibodies targeting the phage cells before they have time to target the phage cells, have time to target the bacteria.
So his question was, is there ever a problem with the phage, the bacteriophage, being targeted by the human immune system before being targeted by or before they can get to the cell? So bacteriophage themselves aren't actually cells, they're viruses, so they're much smaller. They can be targeted by the human immune system, but they don't actually they're not known to attack human cells. This is why they're so important, is because we could specifically target only bacteria with them. So they can be targeted, but it's not a priority that the immune system would have.
And kind of going off of that, we already have bacteriophage in our body, so we know that it is a compatible relationship, which is why it makes it such a viable option instead of antibiotics.